Proving a new pharmaceutical actually works usually involves massive, incredibly expensive, and really time-consuming human clinical trials. But what if we could just skip them? Well, for a whole class of really complex drugs called biosimilars, the European Medicines Agency is literally rewriting the rulebook to do exactly that. Welcome to this explainer, where we are going to unpack the EMA's landmark 2026 reflection paper. We're going to see exactly how skipping these massive comparative efficacy trials could mean dramatically faster, cheaper access to life-saving medicines.
Okay, let's take a quick look at our roadmap for this explainer. We're going to cover 1. The paradigm shift, 2. The precedents, 3. Rules for waiving trials, 4. Justifying quality attributes, and finally, 5. The clinical perspective.
All right, let's jump right into Section 1, the paradigm shift.
So, do we always need human trials to prove a biosimilar works? This is the million-dollar question, right? Historically, the answer was a resounding absolute yes. You needed a comparative efficacy study, or a CES, to prove a biosimilar candidate performed exactly like the original reference medicinal product. But the EMA has looked back at years of regulatory experience, and they've definitively stated that these comparative efficacy studies, well, they often just don't add essential scientific knowledge anymore. I mean, when you have today's state-of-the-art analytical tools running hundreds of patients through a massive clinical trial just to confirm what you already proved in the lab, it's kind of becoming redundant.
And that brings us perfectly to the newly adopted EMA reflection paper, which actually outlines how developers can waive these efficacy trials altogether. This document is a huge deal. It finalizes a regulatory pathway where you can confidently infer similar clinical efficacy and safety just from incredibly detailed physicochemical and functional testing paired with some targeted pharmacokinetic data.
Now, why does this work? It all comes down to a really elegant, universal scientific principle. Structure determines function. The biological activity of any active substance, whether it's something simple like paracetamol or a massively complex monoclonal antibody, stems entirely from how it interacts with its target receptors. So if two complex proteins share a highly similar structure, they are absolutely going to bind to the exact same receptors in the exact same quantitative manner. And as a direct result of that, they're going to have the exact same clinical efficacy. It's essentially just physics and chemistry driving clinical biology.
Moving right along to Section 2, the precedent.
Now, you might be understandably skeptical about just skipping human efficacy trials. No human trials? Really? But this actually builds on a solid concept called comparability, which was established about 30 years ago by the ICH Q5E guideline. The whole concept of comparability recognizes that biologicals are inherently variable, right? Literally no two batches are ever perfectly identical. Comparability says that minor differences in quality attributes are totally fine, as long as those differences absolutely do not translate into adverse impacts on clinical safety or efficacy. It's all about proving that our existing knowledge makes the analytical data incredibly predictive of actual human outcomes.
Just look at the stark contrast here between the historical gold standard and this modern tailored pathway. We are fundamentally shifting away from relying on those expensive, mandatory comparative efficacy studies. Instead, we're relying on state-of-the-art analytical and functional characterization. The EMA is basically saying that the analytical tools we have today are actually far more sensitive at detecting differences between a biosimilar and its reference product than a blunt clinical trial ever could be.
And to prove this isn't just some abstract theory, we have actually been doing this for years behind the scenes. Think about it. Since the 1990s, if a pharmaceutical company wanted to make a major manufacturing change to a biological drug, like, say, completely replacing a master cell bank or changing their expression system, they didn't have to run a brand new clinical trial. No way. Those changes were routinely approved using just analytical comparability data. From a scientific viewpoint, making a major manufacturing change is exactly the same scenario as developing a biosimilar.
Okay, let's get into the specifics with Section 3, Rules for Waiving Trials.
Regulators aren't just handing these waivers out to anyone. To even be considered to skip comparative efficacy trials, developers must hit every single one of these 5 prerequisites. First up, you have to comprehensively understand the drug's mechanism of action, or MOA. Second, you must use state-of-the-art orthogonal analytical methods. That basically means you have to look at the molecule from completely different angles using totally independent testing techniques, just to ensure absolutely nothing is hiding in your blind spots. Third, you need incredibly robust functional assays, in vitro pharmacology, to prove the drug acts the exact same way in a test tube as it would in a human body. Fourth, you need a validated manufacturing process for batch-to-batch consistency. And finally, you have to have a pre-established similarity assessment plan agreed upon with regulators before you even start.
To pull this whole thing off, the EMA recommends testing 15 to 30 batches of the reference medicinal product.
Now, why so many? Well, to establish what is actually highly similar, you first have to understand the natural baseline variability of the original reference drug that's already on the market. Testing 15 to 30 batches gives developers the data they need to properly estimate that real batch-to-batch variability, so they can set precise similarity ranges for their own biosimilar.
All right, Section 4. Justifying quality attributes.
The EMA is crystal clear here. When inevitable, tiny molecular differences pop up during testing, they absolutely must be scientifically justified. And as you can see, different anomalies are handled differently. While some variations, like charge variants, can be pretty easily explained away with some focused lab work, others, like glycosylation, which is how sugars are attached to the protein, require highly complex functional justification to prove they won't impact the drug's safety or efficacy.
Take charge variants, for example. It's actually quite common to see differences in something called C-terminal lysine clipping. I know, it sounds super intimidating, but because scientific literature already proves this specific modification doesn't impact safety, developers can basically solve this analytical puzzle using specific enzyme treatments. They use things like carboxypeptidase to isolate and neatly explain the variants. Easy enough?
However, if you find differences in specific sugar structures, like afucosylation or high mannose, well, that is a much bigger deal, especially for monoclonal antibodies that rely on activating the immune system. To justify these differences, developers have to follow these precise steps. They must run comprehensive receptor binding assays and run ADCC assays on actual human immune cells. They basically have to establish hard, undeniable correlation data proving those sugar differences do not alter the drug's biological activity. If there are clear differences in how the biosimilar triggers those immune responses compared to the reference product, the waiver will be rejected outright and approval might not even be possible without going back and altering the manufacturing process.
And that brings us to our final section. Section 5. The clinical perspective.
So, putting my clinical researcher hat on for a second, despite everything we've talked about with waiving trials, human patient data isn't entirely gone from the equation. If a biosimilar candidate falls into any of these categories, that analytics first approach is thrown right out the window, and comparative efficacy studies are absolutely still required. For instance, if the mechanism of action is poorly understood, which happens a lot with complex cell-based products, you just can't confidently use analytics as a surrogate for clinical outcomes. Or maybe your analytical methods just aren't sensitive enough yet. Furthermore, if the drug is applied locally, like a topical cream or an eye drop where it doesn't absorb systemically, you can't measure meaningful pharmacokinetics in the bloodstream. In all those cases, you have to run traditional clinical trials. No exceptions.
And there is an absolutely critical caveat to all of this tailored approach stuff. Even when a massive efficacy trial is successfully waived, the EMA mandates that comparative pharmacokinetic or PK studies are still essential. You still have to actually give the biosimilar to humans, usually healthy volunteers, to definitively prove that the human body absorbs, distributes, and clears it at the exact same rate and concentration as the original drug.
And here's a really cool, brilliant bonus to those required PK studies. We also rely on these smaller human trials to monitor safety and immunogenicity. We're specifically looking for anti-drug antibodies, or ADAs, which is where the patient's immune system attacks the drug. Regulatory experience over the last decade has proven something amazing. The immunogenicity data collected during these relatively small, targeted PK studies tracks just as accurately as the data observed in the massive, old-school efficacy trials. It turns out we can get the vital safety signals we absolutely need without subjecting hundreds or thousands of patients to redundant testing. It's a win-win.
Ultimately, the EMA's tailored approach acknowledges a fundamental truth. Modern analytics often provide far more precise guarantees than blunt clinical trials ever could. So I'm going to leave you with this to chew on. As our technologies perfect their ability to literally map every single atom of a drug, what other massive human clinical trials will soon become completely obsolete? Keep your eyes on the science, folks. It is literally reshaping the future of medicine right in front of us.